All right. Good morning, afternoon, evening, wherever you may be, folks. Welcome to Harvard University's Microbial Sciences Initiative Summer Seminar Series. Our goal with this summer seminar series was to bring you a variety of speakers who do work that in the area of microbial sciences that just is really exciting and inspiring. And we've heard from a couple of great folks so far. And today we have an equally outstanding speaker who is my friend and colleague, Dr. Mark Martin. And it's really my great pleasure to introduce Dr. Mark. A lot of folks call him Doc Martin. Mark is a treasured member of the microbial community, of the microbiology community and microbial community. They probably love him too. He's one of the most innovative and inspired of microbiology educators because he stays atop the literature through his own scholarship and his own research and tailors the undergraduate experiences to ensure that his students are also at the leading edge of the field. Now, Mark earned his bachelor's degree in biology at UCLA, my alma mater. Go Bruins! And then a PhD in biological sciences at Stanford University. After that, he did postdoctoral research at UC San Diego and then worked in the biotech industry for nearly 10 years. Notably, Doc Martin worked on bacterial polymers used in foods like xanthan gums, which show up in salad dressings, and gellan gums. Am I pronouncing that right there, Doc Martin? Is it gellan?
Gellan, yeah.
Gellan gums and things like Snackwell cakes, for which I'm embarrassed to say I have a very soft spot for the Snackwell's devil food cookies. But Mark then returned to academia and brought with him a wealth of knowledge and experience from both academia and industry. Now, Mark likes to say that he chemotactically arrived as a tenured biology professor in the Pacific Northwest, teaching courses on microbiology, symbiosis, and introductory cell and molecular biology. His classroom focus is both conceptual and, I love this, ownership-based, because Mark encourages creative science art in its myriad forms to encourage engagement, ownership, and accomplishment. In his lab, Mark studies bacterial predators, bioluminescence, and symbiosis, and his lab is a totally proven incubator of success for students, as Mark has sent 24 students to PhD programs, and 5 of them now are currently in faculty positions. And to me, or rather to Mark, I should say, individual student success is everything. Individual students and their success are everything. That's a phrase that Mark embraces and a phrase that I greatly admire. And it's that conviction that earned Mark the ASM Karski Award for Distinguished Teaching in 2018 and currently an ASM Distinguished Lectureship starting in 2019 and going through 2021. And I'll just end by saying that Doc Martin and his wife, Dr. Quinn, are very proficient in the art of painting. Although their media is quite unconventional. And I'm going to leave it at that, and I'll let the good doctor tell you about all of that stuff himself. So without any further ado, we'll be welcoming Doc. Oh, let me actually say one more thing. While we are here live on YouTube, please feel free to use the chat box to post your questions for the good doctor as he is talking. That'll give us a chance to go back to the questions, and Dr. Hannah Gavin will pick this up after Doc Martin gets his presentation, gives his presentation, and we'll take the questions you post there in the chat room on YouTube Live and share them with Doc Martin. So now, without any further ado, let's welcome Doc Martin. Doc Martin, the screen is yours.
Thank you very much, Pete, and welcome everyone. I'm now going to share my screen. Let's see if things go away when I do that. You know, it's always a learning experience, isn't it? But now you should see this and we should be on our way. My goal today is to talk to you a little bit about how to teach students to see the world with microbial eyes. And I want to explain what that means as we go. But before I get there, I just wanted to say that it's a pleasure to be here virtually. I've only been to Cambridge once. So at some point in the future, when our current crisis has abated, hopefully, I'll be able to come visit again. But until that time, I wanted to say one of the really excellent things that I've noticed is you have an interesting history of microbiology at Harvard. For example, this gentleman. is the late Dr. William Augustus Hinton, who is a true microbial hero to me. By the way, this is painted by my wife, Dr. Jennifer Quinn, using bioluminescent bacteria, and I took the photograph in the dark. I spend a lot of time in the dark when you think about it. Now, Dr. Hinton was the first Black professor at Harvard Medical School, wrote a textbook, was a wonderful immunologist and microbiologist working specifically on syphilis, and father to Dr. Jane Hinton, who was the co-inventor of Mueller-Hinton media that most of us use in the lab. So if you haven't gone to see, I believe there's a wonderful room in Harvard Medical School that celebrates Dr. Hinton. Please go there and lift a glass to him for me. A remarkable man and a remarkable life. So with that, I normally, whenever I give talks, I bring buttons and stickers. And more recently, I brought 3D printed tardigrades with me. And I am so incredibly sorry that I can't distribute that kind of microbial swag to you today. COVID-19 forbids. So my apologies. And I mean that heartfelt. I so enjoy kind of promoting microbial PR. And now I have to do it virtually. So I'd like to take you on a microbial journey if I can. The center of my philosophy with students would be one thing I'd like to discuss with you. And now I have to go back here. Sorry about that. How I became obsessed with what I call matters microbial. And I apologize for #mania. And then the issues that I see with microbial PR. And this is prior to COVID-19. You can imagine how challenging our time is now. Strategies I use in my classroom to promote an idea I call microbial centricity. And the most valuable thing that I do is to coach my students to success in the sciences. We all talk about how much we hate grading when we're educators. The payoff is when you see the success of the individual student. So what do I mean by seeing through microbial eyes? Well, I try very carefully when I show art to give a shout out to the artists involved. This, for example, is something the Amoeba sisters, if you don't know who they are, you should, put out. Growing up when and where I did, the whole idea of me actually quoting Proust is kind of unusual. And in fact, this is something I came to some time ago on the Elio Schechter's blog, Small Things Considered, about this idea of not having new landscape, but new eyes. And that's what I try to do. The two central concepts I bring, not just to micro, but to all of my classrooms, is about the beginner's mind. I had an interest at UCLA in Zen Buddhism, and the idea of the beginner's mind is you don't have preconceptions. And you're able to come up with interesting ideas that no one told you were bad ideas. If you sit and think about it, the number of times that you've had a good idea, you expressed it, someone said it wouldn't work, and they were wrong, are numerous. I'm not saying try everything, but I am saying follow your gut on these things, microbiota accepted. And I think Will Rogers makes a good point, as always, and we see this in the media today. It's not what we don't know that gets us in trouble, but it's what we know that ain't so. So in order to kind of really look at microbiology and look at the world in a fair-minded fashion, full of joy for the new things that we see. So my take-home lesson to all of you at this early stage is very simple: to never, never quit. I made so many errors and mistakes, but I never gave up. And to be honest with you, I have no idea why I didn't, but I did hang in there. I refused to give up. And I cringe at myself in my 20s. I was so beyond awkward. But people can grow up and change. And this is why I always tell everyone in my classroom and during talks, we need to be kinder to one another, to listen more, to judge less, because everyone is on a journey of their own. Getting back to the classroom, or the lab in my case, mentoring is crucial, especially for first-generation students like myself. I had no idea how any of this worked. Academia can be insular and exclusionary, and we need to make it more open and welcoming to people from all walks of life, all faces, all voices. Science belongs to humanity. So what I would tell you, and I've been surprised, I've said this for the last few years, the number of people who privately tell me how much this means to them. Be authentic, be genuine, and be yourself. So as you might guess, and I'm actually toned down today, I'm somewhat flamboyant. And it used to worry me, and it certainly has cost me quite a bit over the years. But I believe that students need you to be genuine. They need you to be who you are. So I do lots of silly stuff, as you can see. You be you is a motto that I encourage everyone to take. Students notice, it gives them courage. It gives them the freedom to ask you questions they might be afraid to ask. That's, my wife made me that mask, which I love. And lots of people have made like pieces of art. I have absolutely no artistic talent, by the way, but I know artists. And I simply love what I do with students. There's no substitute for it. So the students need your enthusiasm and your passion. So you need to give it to them. Don't be afraid of it. And you will have people telling you that you shouldn't do that. And you need to follow what you believe. So I tried to brand myself years ago. At my institution, we have a marine biologist now that we just hired, wonderful person who does some microbiology. So I'm not quite the only one anymore, but I still have to represent. And many years ago, a student made me this, and that involved the P word that Norm Pace doesn't like so much. And so I had to kind of get rid of that over the years. And then I changed my tactics a little bit, and I came up with this idea. I had someone in a faculty meeting call me a microbial supremacist, which at the time was funny, is not funny today. So for a while, I had that as a motto until that word supremacy became uncomfortable for many people, and rightfully so. So I found a way to change it again. So microbial centricity is my watchword now. Now, this is important. You need to be flexible. Times, people, society change, but our passion for what we do does not. Be open, be genuine, be honest, and be kind. We need to preach microbial centricity early and often in the curriculum. I know several people, Margaret McFall-Nye and Diane Newman, have tried to get together to figure out ways to introduce more microbial science into introductory biology. I've tried to do my own part on a much smaller scale. So the most important take-home lessons is we need to occupy the curriculum. And yeah, there's that awful S word up there again, because that's an old piece of art. But we really need to put more of it in there. It's great for students because experiments can be easier and you can make them relevant in lots of ways. But I had my work cut out for me. I really did. I believe that microbes get really bad PR often. And so what can you do about it? And this is what happened when I asked some first-year students their response to the word bacteria. And as you can see from this, there's a great deal of negativity here. And when I asked my junior and senior level microbiology students the same thing, again, now these are people who are juniors and seniors, I still got a lot of negativity to it. So we have work to do, all of us, to promote microbial centricity, to create good microbial PR. And then this happened. And this makes bad microbial PR even worse. So we have to work so hard to keep this from occupying our minds and our students' minds. So the true microbial truth, well, too many truths, but you know what I mean, is that most people have always portrayed microbes as always bad, as devil microbes, or, and this is not so good either, always beneficial, angel microbes. But the real truth of the matter is that most microbes don't care about us. We're narcissistic and we're concerned with ourselves, and that's not a bad thing. But we tend to forget that the vast majority of the microbial world has nothing at all to do with us, period. And so when you teach microbiology, that's an important point to keep driving home. So how do I bring these microbial truths to my classroom? Well, I actually often ask students to go looking in the media for examples of this. And it's what you can see, lots of students have done this for me in the past. They've come up with things they've seen that they felt portrayed microbes in a negative sense. And also there's the issue of the common swab story. We've all seen them where someone takes a swab, they swab a surface and say, yo presto, there are microbes on it. Well, of course there are. I call those swab stories. Now you have to remember now in our new reality, this is going to be much more complicated, but it's still important to drive the point home with students. So I try and proselytize a microbial perspective every day to get past that yuck factor, So this is a piece of artwork a friend of mine made some time ago, and students invariably get all grossed out by the idea. Even though we're all socially distanced and sanitized right now, you still get the point. Yet I encourage them to think not just about handshakes, but more personal things. And we have no trouble with those. So it's a matter of perspective, and it's important to kind of create that microbial perspective. I also like to show them to some degree the kinds of things that we see in their water bottles. I call them water bottle buddies. And sometimes we get interesting images when that happens, but mostly it's an idea to show them that even in their reusable water bottles, we tend to see microbes no matter what. Doesn't matter if you wash them, doesn't matter if you scrub them out, because they're part of us. We're afloat, aswim, moving through a sea of microbes at all times. And the ones that are bad are like the juvenile delinquents of the microbial world. They're very small in number, but they get all the bad press, don't they? So I like to bring a sense of community, and this is what I've been doing for years with buttons. I even make microbial candy, custom made, I don't make it. And then I make these t-shirts for students over the years. I don't do this in any way to waste money. I do it to promote an idea of microbial perspective. And this idea of bringing people together in a group works and is very positive. Lots of artwork becomes obvious, and things that we do to promote these over the years. Lots of artists have worked with me. I've worked with pigmented bacteria to try and create this idea. There are lots and lots of ways to kind of keep this going. Now, I want to point out that some people might look at some of this and might be claiming that in some ways, this could seem like it trivializes. I disagree. I believe very strongly that things that stick out in your brain are remembered. And because of that, these are the things that students will remember and hopefully will help them as they navigate through our increasingly challenging world. I also like to use what I call microbial shock and awe. It's funny to show them what's in their water bottle, and they always get amazed by the kinds of things that they see. We often streak some of these to singles, and we do 16S identification to find out what's there. And it's been so interesting, the kinds of things that we've found. And in fact, many of the students come to think of them as like their personal friends, which I find very interesting. So those are the water bottle buddies that we talk about a lot. Nothing would please me more than to see a nationwide network of students doing this, because there's so much interesting stuff that happens. I also like to talk a little bit about how we take things for granted in microbiology, which brings me back to G. Osmond. And let me see if I can play this for you.
That smells like dirt. No, dirt smells like streptomyces.
And this is true. And it's something that I promise you every student that participates will not forget. In addition, I often discuss the issues that microbes have to survive in the environment. Pseudomonas syringae makes a protein called ice nucleation protein that can cause the very rapid conversion of chilled water to ice. And Joe Handelsman taught me this some years ago, but it is always a winner with students.
All right, 1, 2, 3. Wow.
That last laugh made it for me. And now I have to go back whenever I do that. There we go. So what's important here is engagement and ownership. We want to actually have things the students are physically doing, and we want them to feel that they own it, that they feel some connection to what they're doing. To be passive, to sit and listen, is the enemy, which is our problem in the coming semester and year. I often bring up some interesting aspect of microbes to approach in each lecture. I call it a microbial minute. Otherwise, folks are just leafing through textbooks. That's what I did as an undergrad. And in fact, there are better ways to show them the depth and breadth and sheer wonder of the microbial world. The community building works, as I've mentioned before. All assignments need to be cleared, scaffolded, and there should be a very clear rubric. You don't want surprises. You want students to feel that you're working with them on it. I actually have them write not just a term paper, which I call a microbiography, but actually a one-page version, which I call a nanobiography, which is supposed to be for their English major friends, because I believe the idea of communicating science effectively to non-scientists is very important. We also have small research projects, or we would have, and those really take off for students. It's a chance for them to investigate for several weeks something of interesting to them. And then finally, some kind of creative approach. Not everybody wants to do them. And I am amazed the number of times that students tell me that they lack talent. None of that's true. They have fabulous talent, and I can show you in a moment. So some of the independent research projects students have done, they have to be individualized, and it's really tough. You have to run around crazy, but it's so worth it to see the look on the students' faces. For example, when you actually do leaf prints and are able to show the students, or have them show you, which is better, pink pigmented facultative methylotrophs that are living off the methanol and the exhalations from plants. Agar degraders from seawater, which are wonderful. Looking at stress versus colonial variation in Serratia, asking them how those colonies form, get them together in groups and say what had to have happened with the expression of those prodigiosum genes. And then finally, everyone likes bioluminescence. I certainly do. And thanks to Heidi Goodrich Blair, I have a strain of Photorhabdus that we can infect waxworms with, and you can actually see these in the dark, which is wonderful. Now, students will surprise you if you give them the option of reaching out for their new stuff. So here's something fun to try. Ask students on an exam to draw a cartoon depicting a concept from lecture. Tell them you're going to, don't spring it on them, tell them you're going to do that. True, some people will copy things that they've seen on the internet. I guess that's okay, but you'll see other things. Like this. Microbial tinder. What on earth do I do with this? I don't even want to talk about some of the stuff here, but it's funny. Or this one, which is much more serious, actually understanding how quorum quenching works in a cartoon. We actually can see a particular bacterium intercepting a message meant for another. And from my first year class, one of my favorites, why G proteins are called G proteins. And I have no trouble giving a point for humor. So my point is, this is their creativity, not yours. And they're sharing it with you. So I've also asked my students to come up with microbial haikus over the years about information in class. And here are two great examples. Look at these. They're brilliant. And these are students who are generally pretty quiet in class, don't have much to say, but you give them the opportunity and look at the wonderful work that they do. I believe that most students can do things like this. We just have to give them the opportunity. And finally, I like to give them creative projects. Again, they have to come up with them. So students have created magazines for me. They've painted things, the famous Minot quote. I have wood blocks of a type 6 secretory system and Penibacillus colony morphology, and a piece of ceramic that just knocked me over to see. And even from last year, wonderful work from students. This student I had no idea was a cartoonist, but look at that wonderful work. This is discussing how type 6 secretory systems are working with Vibrio fischeri and its symbiosis. And then many students interested in making cross stitches of various evolutionary ideas. This is fabulous. And then finally, one of my favorites, a student built me a flagellum out of pipette tips. Now I have to tell you, I bought them the pipette tips for this, but I promise you, because I asked it on the final exam, this student completely understood the structure of a flagellum because they owned it. And I think I want to say that for the quieter students, this is really important. I had a very, very quiet student years ago, soft-spoken, shy, nearly silent in class. And this is what she did. And this doesn't come up with a picture very well. Look at this, folks. This is the part that knocks me out. You have to appeal to and approach and support the individual. And it's hard to do in big classes, but anything you can do will help. You have no idea the kinds of paths that students have taken and how hungry they are for your paying attention to them. And then, of course, you've already heard some references and seen some Lux art. This is a strain of Photobacterium legionothae that was isolated by Ned Ruby and Eric Stabb in Kaneohe Bay in Hawaii. And it's extremely bright in my hands. And if you see the kind of work that Dr. Jennifer Quinn does, here's another microbial hero, Carl Woese, but not just them. I will have students say that they have no skills, but look at the stuff that this was a first-year student, this was a senior, and this again was a first-year student. So in my class, when we're present, we often have the Lux Academy Awards every year, because I really think it teaches two issues. It teaches that microbes aren't necessarily bad, and it kind of scratches that artistic itch, and it pulls home different ideas. So really quickly, I tend to give these, we might be short on time, so I want to be careful here, but I put on workshops. And this is at Cal State Sacramento, just to show you some of the work. So you get the idea, friends, that what we've got here is an idea where students can do fabulous work trying to express themselves through this issue of art that kind of, as I said before, shows that microbes are not necessarily bad, again, reaches that connection between art and science. And even leads them to things like quorum sensing and the idea of the enzymology of luciferase, things of that nature. I've posted a lot of these things on YouTube. I don't mean to take up your time with it now, but I so enjoy going to places and putting on these shows because what the students produce is so wonderful. I also want to talk about Mariko, a student I had years ago. And Mariko used to write cartoons on her exams like this. And at the end of the course, and I didn't ask her to, she made this. And these are all things that I had talked about in class. I used to make a joke that students were petri dishes with shoes, and she found that amusing. But what was interesting is that she took a term I used, micronauts. This is what I like to call my students in microbiology, micronauts. And in fact, she said, micronauts we soar with a little rocket ship, and that became a fancy pin and a motto. And I have to tell you that never would have happened if I hadn't listened and worked with and appreciated and supported different types of students with different ideas. It's often said that 10% of your students will do great no matter what, 10% will have trouble no matter what, and we don't put enough time on that 80%. And this is important because those 80% have wonderful ideas. I do a small amount of research in my lab, as Pete has told you. It's like a boot camp for graduate school, and we have buttons and shirts and hats, and we do a little work in the academic year, but classes make that difficult. But we work really hard in the summer. But the learning and training is the first-rate issue. That's what I care about. I want every student coming out of my lab to be a real scientist in their outlook. So this idea of community, this idea of support, and this idea of critical thinking are really important to me. And the results speak for themselves. So when I look at students that have spent more than 1 year in my lab, I have, actually, I believe it's 6 now, faculty members across the country, 24 have or are in PhD programs, 6 in MD programs or with MDs, 3 in PA programs, 5 research technicians. So what I call the one microbial truth is a joke actually works. They're all fabulous. And I am not a great teacher and I am not a great researcher, but what I am unreservedly is a good talent scout. So things I've learned about creating lifelong micronauts. You be you, the bench, the lectern, the blackboard. Students will recognize and respect your authenticity. Express your passion for your subject. Do it honestly. Be who you are, of course, but be honest about it because students recognize it and crave it. Be clear, be organized, and as fair as possible. It will not be perfect. You will make mistakes. Lord knows I do. But do the best you can because you need to honor student ownership, engagement, and authenticity. Don't be afraid to show them. I often tell them when I was 11 years old, the first time when I looked at a plate with photobacterium on it and how it rocked my world in a way that I can't really express. And they laugh at first, but then later they say that they were really glad they heard that. So to finish up, because I know we want to have some questions and maybe other things that folks want to talk about, I want to thank Pete for the invitation to speak with you today. Also, Hannah and Julia for all their positivity and technical support. I want to thank the ASM and the Karski Foundation for supporting my a little bit odd view of microbiology education. NSF for inexplicably funding some of my research. The Murdoch Charitable Trust, again, for inexplicably funding some of my research. And the University of Puget Sound for allowing me to support my students to do research. And my wife and children for everything there is, and all of you, as we disperse today, this quality quorum. Never forget we're on Team Micro. Keep your mind on your microbes and your microbes on your mind, to misquote Snoop Dogg. And finally, thanks to Harvard. And I will now stop sharing.
That's been awesome. Thank you so much, Mark.
I was glad to be here.
We'll send you some virtual applause here to start, and we can get started with the Q&A. For anyone who's around, you're welcome to submit questions that you think of as we go. I have one that came to mind while you were talking, and I'm curious when you were mentioning the idea of the beginner's mind.
Yes.
How you promote this in areas where you're knowledgeable. So do you often feel the need to continue expanding to new topics so that you yourself can adopt that beginner's mind? Or is it something that even in areas where you feel really experienced in microbiology, you're able to promote in your students? And if so, how do you continue to do that?
So Hannah, I have a whole story about this, but because I talk so much, I didn't put it in there. So I'll give you the short version now. The first postdoctoral seminar I ever gave was on bioluminescence. And it was in the old days, back when dinosaurs ruled the earth. I had a 2-liter Fernback flask. I had a small amount of culture. I had the lights down and I gave it a big swirl and held it up next to my ugly mug to illustrate. Now, what's important here is that there were about 50 people in the audience and a high school student. And the high school student didn't know that you weren't really supposed to ask questions in the middle of a seminar. That's changed a little bit, but back then you never did that. So his hand went up and I called on him. Now, mind you, I had studied as hard as I could about how luciferase worked and how bacterial bioluminescence worked. And I was by no means an expert, but I knew a lot of stuff. And I called on him because I'm not threatened by it. He had a question. And he said, so I can't see through that flask in the light. And I said, that's true, because it's scattering the light. That's why it looks opaque to you. He says, so how does the light get out? And I sat down and I thought about it. And I told him, I've never been asked a question like that. And it's a fabulous question. It's a question where you didn't think, is that a good question? You just asked it. All I can tell you, and it's a guess, is I want you to imagine each bacterium like a lampshade with a light bulb inside. When the lights are off, you can't see through all the lampshades. But when you turn the lights on, the light shines from outside. And he said, that makes sense. And that's an example of a beginner's question. And so I tell that story, and then I encourage students to ask things like that. Now, we always have a balance. If you get asked too many questions, you aren't able to cover what you want. But I honestly think that if one person has a question, at least 3 or 4 other people do. Mm-hmm. So, try to answer it in that way. I have no trouble saying the 3 most important words in science are I don't know. And many, many academics don't like saying that because they feel they won't be respected. I don't think that's true at all. I think when you put it out there, it becomes collaborative. That too long of an answer?
No, that was perfect. Thank you. We have another question here. Someone asking, how do you balance the material you present so that you're reaching everyone in your classroom? How and tying that into how to hit the right level of complexity or detail?
This is such an important question. At a large institution, where I went actually as an undergrad, I wasn't a microbiology major at UCLA, but I took lots of microbiology classes there. And so when I took introductory microbiology, we didn't spend a lot of time on physiology. June Lascelles taught me physiology later. Bob Romig taught me virology later, that kind of thing. So for an institution like my own, where there's one microbiology class, how do you do that? And all I can say is you have to pitch to levels. In other words, you have to ask yourself, what do you want them to pack into their lunchbox? The stuff that they take afterwards. I've had some people say to me things like, students can look up whatever they want. That might be true, but if you're learning a language, there's a certain basic vocabulary you need to have. And so that's going to be different for each institution. But you do have to be very, very careful to find ways to reach out to and assist people who might not have had prior background about these things. So, for example, when I talk about quorum sensing, there's a level of that I teach that every student's going to be able to get, and then we can get more complicated on top of it. But at least I know that they have that basic that they wouldn't have had in any other class. In addition, I was able, after beating, you probably see the wrong side. Oof, I can't do this backwards. I give up. You notice where it says bang your head against the wall? I finally, after many years of kind of workshopping this in my microbiology class, got a biofilm lab in our introductory curriculum. And, I pitched that in terms of data analysis and all the rest. And so they were able to measure that. And that is one way that you can take, here's a student who could care less about microbiology, but they understand the basic ideas of it. But it is indeed a moving target. And so I want to thank that person for that question.
Definitely. We have one here. How do you get yourself amped to teach material you may not be as excited about?
Well, this is a big problem because for many years, I thought I had a poker face, and this is a lie. Whatever I'm feeling, look at Pete laugh. Everything I'm feeling shows up on my face. And so I have to be really careful about that. And there's like, you may have had this impression a little bit, well, a whole lot of a smart aleck inside of me, and you have to really watch that. Depending on your institution, depending on your colleagues, depending on your students. But the most important thing is reaching this commonality with them. So, for example, I am not a big fan of metabolic, well, that's not true. I do like some metabolic pathways, but when I'm teaching basic metabolic pathways, it doesn't excite me. So I sit down and I think, okay, what do they need to pack into their lunchbox of information? What do they have to know? And not just the idea of what I used to call the bug parade, where you learn all these factoids about microbes. So if you're not going to use the EMP pathway for glycolysis and you're going to use entner-Dodorov, now that's different. What organism uses that and why? Does that make sense? In other words, can I tie it to something?
Mm.
Connected to that is the idea, what about something that really excites you? For me, it's small RNAs. Students get frightened. And so you really have to watch yourself not to go too far on it.
Mm.
I've gotten so, here I am being too direct. Some of the diagrams that I see commercially available to explain things don't sing with me, so I just have to draw my own. So these are either side of the teeter-totter that we have to deal with. And again, I would remind everyone of that 10%, 10%, 80% issue. You don't want to ignore the people who are going to do great no matter what. I mean, they actually keep me going. And you don't want to ignore the ones who are having trouble. You want to do whatever you can for them to meet them in the middle. But that 80%, those are the ones that we really have to reach. So I have several things that I do with my micro students in particular, where they take a concept in microbiology and have to explain it to someone who isn't a science major, as I've mentioned before. And that seems to help a little bit with centering it.
Oh, that's an awesome idea. This
Thanks.
is a timely question we're getting from the audience. Can you tell us about your experience transitioning to remote and distance learning? What are you finding that still works? And was there something that worked better than you expected it to? So actually, I'd be curious. I know we've talked briefly when we weren't on the seminar about the fact that you're transitioning for fall, but I don't know what happened at Puget Sound for the springtime too. So where are you in that process and how are your experiences going there?
I cannot express to you how challenging, and when I say things like that, many people deal with a million times worse from whatever I'm talking about. So please don't take this as a general thing. But it was very difficult for me. We went to all remote about spring break. So I'd had a few weeks with students in, I was teaching Introduction to Cell and Molecular Biology, which is a critical class. Oh, Lord Almighty, it's so important because this is where you set the stage for everything. This is where students get turned off or turned on to science. This is the stage where they decide they hate science or they love science. And it's so critical. And so all of us in my department are constantly trying to come up with ways to really engage the students. And sometimes we pitch too high, sometimes we pitch too low. So it's always a moving target. And so when we went all remote, all of us had to learn how to do things that I didn't know how to do. I was fortunate in that my wife, Dr. Jennifer Quinn, teaches at an institution in town that has a lot of older students who have jobs, students who are parents, and they can't always make it to class. So she had done what I would almost call a hybrid sort of approach for the past 2 years. So because of her help, I was able to use Zoom effectively to give lectures. The problem I had was with the labs.
Right.
Some of my colleagues would try and we didn't know how often we were supposed to be around. Everything was up in the air. We couldn't tell.
Mm-hmm.
And so one of my colleagues was making videos of that person doing some of the experiments. And so I took a different approach to that myself. And what I did is I looked at each lab that they were going to be missing, and I gave a lecture to them about what they needed to know about it, what they would have seen based on prior things. I'll give you an example. You're familiar with PTC paper that some people taste as bitter.
Mm-hmm.
So we have a lab where you amplify your own DNA and you digest with a particular enzyme and you get 2 fragments versus 1, that kind of thing. And so I was able to get the take-home lessons to them that way, instead of just using other people's videos that you can find online. But to a person, they were really interested or really disappointed they weren't able to find out for themselves. So maybe that was a good approach, that they were still really interested in it. At the same time, I think it's really important when you're doing things online to take a step back and say, different people have different personal circumstances at home, and you absolutely have to make them feel included and respected. And so, maybe taking a step back in your expectations, because it's easy to get carried away. I did several things that helped show the students that I really was interested in what happened to them. And the first is I became extra patient. And that's, again, part of being kind. I have three things that I say that are really important in my classroom. And the first is we always presume good intentions unless proven otherwise, that we don't mind read one another, and that we treat one another the way that we would like to be treated. It sounds so trivial, but it makes life so much easier if you can really promote that and model that behavior. So I would ask students to just tell me how they were feeling. I had some of them make memes, since a lot of students in this age group enjoy memes. And some of them were funny and some were heartbreaking. I've posted some of them in the past. I made little videos of them. And was able to share them with my administration because I think it really gave insights into how students were feeling. Some felt really down, some felt really up. But I have one more thing to say, that I did office hours Monday through Friday from 6 to 7 p.m. and online. And I had very few students talk to me. And I felt badly about that. And I would say, when I would teach, I'd say, I am there for you. And then I had 4 or 5 students write to me and they said, we're sorry we're not calling in, but it means a lot that you're there.
Yeah.
I don't mean to minimize anything. I don't mean to, do any of this. I do mean to say a lot of people felt isolated. And that business of reaching out, however you can do it, is so critical. So to circle back on your question, it's very difficult to teach labs online. There are people who are good at it and do a wonderful job. My experience has not been that positive with it, but I found something that worked. Again, no disrespect to other approaches. Being extra available. It's kind of funny, I work more now than I did before, is helpful. Trying to find ways to treat them as the individual, because there's so much a feeling of isolation and sometimes a great deal of anger, and just to be there to listen to it's important. Giving them opportunities to express themselves, and mostly just not giving up.
Yeah. That's really helpful. Thank you. We got a question about Dr. Martin, you moved to industry and back to academia. Given the COVID-19 academic job market, do you think it's still possible for PhDs to spend a time in industry and then return like you did?
Yeah, I was very lucky. As I've said before, I have no idea. So every time that I feel sorry for myself, and I think we all do from time to time, I remind myself of this. No, it was just a matter of having good connections with friends of mine. I had been working in San Diego and was laid off from, and again, it happens in industry. I was laid off from this job in this biotech company, and I wasn't sorry about it because it wasn't exciting to me. It was a paycheck, but it wasn't exciting to me. And then, you just have to wait. And then a friend of mine who I knew from graduate school wrote to me and said, Mark, I think you should try and go back to teaching. And they really needed someone kind of at the last minute. And then I was able to gain some experience there. And then that gave me enough to be on my feet to become and have a record of teaching to be able to do that. Now, I've been doing this business since about 1994 in academia now. And I've been on lots and lots of committees for hiring people. And what I will say now, and it's really important, is that most institutions will want there to be some experience with teaching. And by that, you want to have some kind of idea about how you approach a classroom, how you monitor, I can't say grading anymore because that sounds awful and sterile, how you evaluate outcomes, whatever it is that you need to do, because those are the kinds of things that we look for. But the most important thing, if you're looking at an institution at a small liberal arts college, is that you have some kind of research program that's something that students can do. Sometimes people pitch above that, sometimes they hitch below. I was never in the market for a job at an R01 institution. And I will say that I've also had friends working recently in business where they're doing fabulous research, and I could easily imagine them making a change. But I would tell you, you just send out a lot of applications, and you have to deal with a lot of rejection. But I think that's true on the job market regardless. And I have no idea what next year is going to be like.
Right. Yeah, it'll be interesting to see how things evolve there. I have one more question that I'm going to ask, and then I do want to protect a few minutes at the end because I promised a few minutes to talk about something that maybe we wouldn't be able to normally have time or space for in a seminar. But we do have a question about how you keep yourself creative in coming up with simple concepts to explain complex ideas and ways to illustrate those complex ideas.
Coffee. No, that's not true. I'm being silly. I don't sleep much, and I worry all the time, and that's not good. So what I will say is that if you get into your rut, it's really hard to be creative. And what I have come to is thinking about what I want the students to come away with. What do they need to know? And often there'll be difficulties. Can I give you an example really quickly?
Sure.
So I try when I'm having my students in lab, in the course lab for micro, I try and help them find something that interests them. And I had a couple of biochemistry students that were all carried away about green fluorescence protein. Mm. And I've used it. I know a little bit about it, but I'm not a biochemist really, which would please all the people at UCLA to hear. But what I will say is that they said to me, couldn't we find, because they're talking about the biorad P-glow plasmid, which is actually has era C associated with it. And I want to salute Gary Wilcox for teaching me about the arabinose operon at UCLA. But it responds to L-arabinose and not D-arabinose. And so they said, do you think we could mutate that gene so it recognizes D-arabinose? So I'm thinking about enantiomers, right? And I racked my brains. How could I do that? What could I come up with? And the best I could come up with is to go to my ancient lab manual by Tom Maniotis about doing mutations in isolated plasmid DNA using nitrous acid, I think is what I used. I couldn't tell you for sure without going back over my notes. So we took the P-glow plasmid, they mutagenized it. I bought them some competent cells to transform it into. And it was really a good learning experience for them because they plated it on D-arabinose and they saw some things that were glowing under the UV light. They were all excited. And then they got really unhappy with me when I said, have you also plated on things with no arabinose? Because in fact, what they had done is just de-repressed the gene.
Oh,
and nowadays, it would be very cheap to actually do, and I believe there's a paper about this. I want to say from the American biology teacher, or maybe JIMBY, the Journal of Molecular Biology and Microbiology Education, about actually sequencing to see what changed. So in other words, and I think it's really exciting when something doesn't work in an unusual way. But students don't generally feel that way. But it was, when they came around to it, they did see that it was really interesting, and they were sad at the time that we couldn't sequence that area. But maybe nowadays that would be possible. So I think honestly, the hard part is to get the students to talk to you where they don't feel they're trying to impress you. They're just talking about just throwing ideas out. And when they do that, I get such inspiration from them. It's really quite amazing. They see things I don't. I have, when we do this thing where I have them swab their water bottles, and I get accused of trying to promote negative microbial PR all the time about it. But what's interesting is you will see some weird stuff there, things that I've never seen. And then you ask them, what does that mean? So for example, they'll find something that's pigmented, and then when they streak it out, it's no longer pigmented. And they'll say, what's this mean? And that's where I started inventing this term, hashtag OMG, for overwhelming microbial greatness, because there's so much we don't know. And that's exciting, we don't know. And once you make them confident that they're not being graded poorly, if they get an odd result. I mean, when I took organic chemistry with Donald J. Cram, by the way, that's quite a name. We were graded on the amount of product we made in OChem, which sounds like Breaking Bad, and that's not at all what they mean. So what I'm trying to tell you is that you have to create this trust, and then they'll come to you with ideas. I also read Small Things Considered every week. I listen to This Week In, fill in the blank, that Vincent Roccagnello puts together. It's really hard to stay connected, especially at an institution like my own. You folks that are at like a big institution have wonderful things. You have people right down the hall to talk to. I have to kind of use social media for that to some degree. Mm-hmm. But staying connected is so important.
Yeah, absolutely. And I love your point about embracing the negative result and seeing where it can go from there, because I do feel like in science, a different result than you expect, it doesn't have to equate to failure. And that's such a good reminder. No, please go ahead.
I was just going to say, I mean, that's the whole thing. This stuff was here before we were ever here. And it's going to be here long after we're gone. It doesn't belong to us, right? So we shouldn't have that kind of buy-in to it. We get to unfold a little bit of what's there. And I have a great, huge poster right outside my lab. It's something that I deeply believe is that there's no feeling like when you have found something out that nobody knew. It doesn't have to be important, but you got to, and it's not like, you found it, you saw it, yours are the first eyes. And there's no feeling like that, no job like that.
Absolutely, absolutely. Is there anything in the last couple of minutes here that you or Pete, if you have, I know you're there and you two know each other well, if you have any questions, something that you'd want to talk about for a couple minutes that is outside of the realm of what you normally get to talk about in a seminar, or Pete, if you have any burning questions in that regard, that would be great.
I don't know if I have any burning questions, but Mark, you are a colorful character, my friend, and it would be really neat to hear, at least I think, about the other things that really inspire you and the way in which your love of microbiology maybe spills over or stitches into other parts of your life. How does this fit into who Doc Martin is? Because being a professor of microbiology is obviously a big part of it, but it's not the only part of it.
Well, I was born in Compton, California, and raised in North Long Beach. And I would say that we were lower middle class, and I pretty much lived at the library. And I fell in love with science early on. Some of my earliest memories are looking through a microscope at Spirogyra under a microscope, and I was looking at Hydra and then thinking about the chlorella algae that live inside. And I did a lot of science fair projects, and I don't want to take up time with that, but I found out something that I didn't know that was really important at the time, and I got all that positive push from that. Long story short, 8th grade, I was teaching planarians, flatworms, to associate the darkness with an electrical shock. Please don't look at me poorly. I thought I had read about how if you took a trained worm and fed it to an untrained worm, it's science fiction boy here, they would learn it. And I found out it's really hard to make mazes for flatworms, but I could teach a conditioned response. Long story short, when I took the trained flatworms and they could remember for long periods of time, and then I cut them in two, the tail half regrew a head, the head half regrew a tail, both sides remembered. Now, I didn't subscribe to Scientific American. I didn't read the literature. I was in the 8th grade. But that was, in that day, that was this big deal about the holistic view of memory being distributed throughout the nervous system. And I had famous scientists write to me saying, you should consider being a scientist because of the science fair. And that changed the way I looked at everything, because I thought, well, I talk a lot, I could maybe become a lawyer. But I've always loved science. It's all I've ever wanted to do. And I like being able to find out new things. I don't know why, but I always have. As far as, everywhere I look, when I go for walks, I'm looking at lichens and mosses. I'm constantly thinking about what takes place beneath the level that we're at. You look at your hand and you look at how rough or smooth your skin looks, and you've got 3 or 4 layers of microbes on there, and it turns out they're doing important things. And I think about this all the time. So maybe there's something wrong in there. I don't know, but it's what I love. And this is what I say to people who want to be scientists. You have to love it, because it's sure not for the money. And it's sure not for the, the easy hours. It's for the excitement. And that sounds weird to someone who's not a scientist, but it's absolutely true. There's a whole world underneath us, and it's always been there. And I'm privileged to be able to look at it. When I look at these electrochemical batteries, these mud watts, and I have one that's been going for 9 or 10 months. And people who work with this, who work with electrical bacteria, they're not surprised by this. But I just sit there, I go visit this thing every day I'm in lab and just watch it blink. And I think about what's taking place in there. So it's true, I read a lot of science fiction growing up, so maybe that's part of it. Is that too long of an answer?
No, I think, thank you for sharing that, Mark. And I hope that those who are watching are inspired and inspire those whom they engage with and then they train to kind of, I guess, I love the idea of being genuine and authentic, because that means that each one of us gets to be the best we that we can be, the best us that we can be, and then the best I that we can be, whatever it may be, the best individual. And bring that to bear on understanding the world around us. I think that's so cool and very inspired. And thank you.
And it won't be perfect. There are going to be people who just don't care for the cut of your jib. But isn't that true everywhere?
Pretty much.
Yeah, absolutely. Well, thank you so much to you, Dr. Martin, and to everybody who's been participating live today. MSI will be continuing our monthly Thursday seminar series throughout the academic year. And so you can keep up with the lineup of speakers for that, as well as other events and news through our website, which is msi.harvard.edu, and through our weekly email newsletter, which you can sign up for on the website if you're not already receiving it, and also on Twitter at Harvard MSI. So that's all for me, and I'll let Pete say any final words if he has those before we sign off.
Not at all. Just thank you, Hannah, and thank you, Julia, and thank you, Doc Martin. It's always a pleasure, my friend, and keep doing the great work that you do, because you inspire all of us.
Thank you so much.
Thank you so much.
Bye, friends.